Welcome everybody to the first edition in 2021 to our webinar series on emerging topics in biomolecular magnetic resonance. Please do not record privately because we anyway record both lectures today and will upload them to the ICMRBS YouTube channel probably by tomorrow or so. Let me remind everybody that we use the Q&A box for asking questions, and you can already put in your questions during the talk once they arise. Then we're basically right away ready for the Q&A when the talk has finished. Alternatively, raise your hand after the talk and we might unmute you there. Today we have two fantastic speakers, Dorothee Kern and Ed Bex, and also two fantastic introducers. So David, it's your turn.
Okay, thank you very much. I don't have any slides, so we don't have to worry about that. It's a great pleasure for me to introduce Dorothee Kern from Brandeis University. Dorothee did her PhD work at Martin Luther University in Halle in what was Eastern Germany, and grew up in East Germany. After her PhD, I was lucky in that she came to Berkeley as a postdoc in my lab and began to work on doing protein structure determination by NMR, and took on a very challenging system which had a number of features of dynamics and enzyme mechanism that weren't anticipated when we started the project. We thought it was a simple system, and it's anything but. And she's worked on that and extensions of it for many years now. So after doing great work in my lab, she moved to Brandeis at the end of the last millennium and fairly quickly became a Howard Hughes Medical Institute investigator there. She's won the Pfizer Award in Enzyme Chemistry and the Dayhoff Award from the Biophysical Society, as well as a number of other major lectureships. I'll say that Dorothee was the most energetic and dynamic postdoc I ever had in my lab. And in her talk, I'm sure you will understand that energetics and dynamics have continued to be a theme of her work in understanding enzymes. So I look forward on hearing the latest and greatest, Dorothee.
Thank you, David, for this very nice introduction. I would say my 3 years in Berkeley in your lab have very much influenced me as a scientist. It was one of my best times in my life, so thank you again. And so what I want to do today is to take you on a time travel to the past and future, to ask a question that has fascinated me since I was an undergrad. How did enzymes evolve to be so powerful? People who know me know that I love power and I love speed. So the evolution of billion-fold rate accelerations in enzyme actually enables our modern life today, and thereby almost all biological processes. So what I wanted to talk today, I thought I will give you two stories. One, to understand how enzymes have evolved over the last 3 billion years to the enzymatic power that we see today. And second, to ask the question, what is the missing piece in enzyme design? Why can we not design really enzymes? And so we will look at the designer enzyme and how this was taken by directed evolution to much faster catalyst. So also you will climb over some mountains today with me, because of course the center of biological function is the full energy intermediates and transition states. So what we really want to do is go beyond these static structures, which are, covering the journal front pages, but rather adding the fourth dimension of structural biology, the time domain. So we really want to look at the enzyme while it's catalyzing and describe the full energy landscape. The first system will be, one of my babies, unlike kinase, which is an enzyme which catalyzes the fully reversible reaction between ATP and ANP to ADP molecules. It is essential for every organism, every cell, So if you knock it out, not a single cell can grow because it maintains the right nucleotide levels in the cell. And here's what I'm talking about. This reaction would take 7,000 years. If you add the enzyme, it's done in 10 milliseconds. One other thing I want to do today is really emphasize why NMR, our love, is actually so powerful to answer these questions. If you take our enzyme, you put it in our NMR tube and add our substrates, this enzyme can actually do its catalytic reaction forever because it's a fully reversible reaction. And with all the beautiful methods, you know, my heroes like, you know, Ed and Art and so on have given us, we can measure the dynamic features of every single atom in the protein, as you're all very well familiar. And so we'll mainly focus on relaxation dispersion experiments today. But the key deal is that the enzyme will actually run through these enzymatic cycles, and we can look at all intermediates and transition states. Methods we're using to have our dentin proteins. So we're not just using NMR, but combining that with a series of different experimental biophysical methods, but also massive computation to finally arrive at these atomic resolution movies. So on our first system, and I will just have a quick introduction because these things are published, what we found years ago by doing these relaxation dispersion experiments, that the rate limiting step is not the chemical step that many enzymologists are focusing on, but it's a large conformational change of 25 angstroms opening and closing. And we found that by looking during turnover at these relaxation curves, and all these red and orange residues could be globally fit to one process, which turned out to be the opening and closing. And if you look at the opening rate, it coincided of how fast its enzyme makes ATP. So it was very early on, right? And so I leave you as an introduction with kind of 3 movies which visualize quickly years of work where they really characterized most step of the enzymatic cycle. And this was really done in combination of NMR experiments, room temperature crystallography, where we see conformational substates, fast kinetics, single molecule FRET experiments, and finally also quantum mechanical calculations, which are not published yet. So just to show you what I want to say is that the rate limiting step, you see this here, is the opening of the enzyme, right? Here my nucleotides bound, a 15 angstrom movement. The chemical step is much faster and therefore very hard to catch. And why is it so fast? You see how the enzyme is perfectly pre-organized, that the phosphate has really just to travel by an angstrom and a half. So all the work is done kind of before the enzyme to align very well the caloric residues to do this phosphorus transfer step. And we have to do quantum mechanical calculations because, of course, you break a bond and make a bond, but also it's not rate limiting. The third thing I just want to mention here is one of the hard things for enzymes to do, and why this enzyme has to close and then to open, is not only to align the active site residues, but to also avoid the energetically favorable reaction, which is hydrolysis. So we live in 55 molar water, and if there would be any hydrolysis, we of course would run through our ATP and we would die. So the way the enzyme does it is by not stripping the water molecules out, but keeping all the water molecules far enough away. And this is in MD simulation, you see all these water molecules as little balls, and then the closest one is these H2O here, keeping them far enough away that they cannot do a nucleophilic attack. Okay, now knowing that this is the rate limiting step, you would think evolution would have worked on making that opening, the conformational change, very efficient. And that's what we did several years ago now, where we asked the question, and that's another fundamental principle I want to bring over. It turns out the energy for all these reactions comes just from thermal agitation, KT. And so in this paper, which I'm not going to talk about, is we found out how enzymes evolved over 3.5 billion years to catalyze these reactions very efficiently, even so we are really much colder today than we were when life arose under kind of volcanoes conditions. And so what we did, back then is really reconstructed all these ancestral proteins, made them in the lab, characterized them, and I just show you here the activity as a teaser, the activity temperature profiles, where of course the low temperature is over here, and sure enough, the old enzymes were really little active at temperatures we have today on Earth, because they didn't live at these high temperatures, at that low temperature, and evolution worked. The enzymes, the modern enzymes, got much faster at lower temperatures. But what I want to talk about today is how they did it, and how they lowered the transition state of this conformational transition. So I told you the rate limiting step is this opening of the enzyme. So after having all these beautiful methods to looking at population interconversion rates with CPMG experiment, what kept me up at night is how can we actually find out the transition state ensemble, because we can't see it because it's not populated. So we're thinking about methods, experimental methods, how we can actually visualize what exactly enzymes do to lower the transition state energy. So macroscopically it's easy. We just turn on the video camera. I will see Julia here at the World Championships running over the mountain. But microscopically, we can't watch it except in computation. So the first thing we did was a targeted molecular dynamics simulation. We know the closed state, we know the open state, and we are actually opening the enzyme. The dilemma is what I show you over here. So we have those structures, but along this pathway, which we make computational, we don't know where the highest mountain is, meaning where the transition state is. And so I came up with this idea that, of course, what we can measure experimentally are rate constants. And rate constants should give us some information about the transition state. So now there are two more tricks I came up with. The first one is what we can measure is a pressure dependence of this opening rate. And of course, pressure we can manipulate by NMR. And so what gives the pressure dependence us? It gives us the difference in the partial molecular volume of the transition state relative to the ground state. Okay, so what we will do now is, but the partial molecular volume is kind of a gross parameter, and I want to get high resolution structures of transition states. So the second idea I had to compare two homologous proteins, one from your gut, and the second one, I'm taking you now into a dive down into deep in the ocean, where organisms live at high pressure. So we will compare two homologous catalytic kinases, one which lives under high pressure and one under low pressure. So here are the first experimental data. Is there a pressure dependence of this conformational change for these enzymes? The mesophilic enzyme, this is now actually, you see Jordan here measure enzyme turnover under high pressure. He's actually cranking physically the pressure up in a little tube. The mesophile has no pressure dependence in this overall catalysis, whereas the pressure enzyme starts very slow and gets much faster at pressures where the organism lives. So now, of course, we wanted to understand this in much more detail and did high pressure NMR. And you're all very familiar that that one can measure NMR spectra to actually near less identical qualities at high pressure than at low pressure, using these little pressure tubes, which were developed by Ronald Patterson and George Warren's lab. So this really worked by Jordan, Renee, and John in my lab. So not only that we wanted to take spectra, but now we wanted to measure the relaxation dispersion for these two proteins as a function of pressure. And what we see, high quality data, which can be globally fit, which give us, of course, the rate of interconversion, but also very importantly, the partial mole volume difference between the ground state and the transition state. And what we see, the mesophilic enzyme basically has the same volume in the ground state in the transition state because it doesn't get much faster, it doesn't really get faster. But what does it mean it's much faster with pressure? What it means is that the partial mole volume of your transition state is actually much smaller than the ground state. And we get very accurate numbers out. It's about 35 mils per mole. Now, I want to convert volume into highly structural information. So the question you have to ask, what defines the volume of a protein? And what defines the volume of a protein is not so much the protein itself, because it's very hard to compress, but it's actually water molecules which solvate charges. And from old, experimental data on model system, the solvation of one charged residues gives you about 10 mils per mole. So looking at these data, we speculated that in the transition state, the piezophile should expose about 3 to 4 charged residues which become solvated by water, whereas the mesophile does not. So now we can go back to our MD simulations and ask the question along these pathways, where do we see charges being solvated? So now I think you're going to start getting dizzy with these wiggling molecules. So I'm freezing it to you. So here I show actually the movie with the water molecules as a little red dots zooming in. And that's another nice thing with MD simulations, you can actually look at the individual water molecules. And what you see indeed for the piezophile, very early in the opening process, do we get solvent molecules coming in to solvate? And we were able to bracket pretty accurately where the transition state ensemble is. It should be where the piezophile becomes solvated, whereas the mesophile does not yet. And why is that? You see that the solvation happens because it opens up, whereas the mesophile cannot open up because the lysine has a very strong Stolz bridge to this aspartate. So there's a ground state and then the transition state. It stays there, whereas the piezophile doesn't have this strong salt bridges, and that's why it becomes solvated in the transition state. Now, there are only 2 residues over here which are actually different. So we had our experiments from the computation. We predicted that this should be about the transition state. Now we can go back to design an experiment to test our hypothesis. And of course, it's very simple. The idea is put those 2 residues into the mesophilic background, and it should become pressure dependence. And that's what we did, and it actually worked, kind of, because this double mutant gives us 30% now of the full pressure dependence of the piezophile. Now, we actually missed one more residue. If we turn around in this interface, there is a 3rd residue which seemed to be responsible for this opening and solvation, which is this valine, which is replaced in the mesophile by the glutamine, again, holding that interface together for no solvation. And that was a very profound experiment what Jordan did. Just these 3 mutations in the entire E. coli protein gave us the full pressure dependence of the E. coli. So now with this kind of experiment, we could visualize by really guiding our MD simulation with our NMR pressure experiments to pretty much narrow down where the transition state is. And just to finish up, it's very different to what every computational experiment, computational paper had, and these are hundreds of papers predicted much earlier in the opening, just when it starts to open, emphasizing why we still need experiments to combine this computation to ask these hard questions. Okay, now the second part, I want to ask, now we have some new knowledge. And what's very frustrating is that we are too stupid to design enzymes, right? You all have seen some of those papers of designer enzymes, but frankly, they are not much better than buffer or some random proteins. And so again, very similar what we did in the first story, we want to ask the question, can nature tell us what's the missing link? So years ago, and this is a really cool collaboration with Don Hilbert. And so what we wanted to do is look at one of these lousy designer enzymes, it's campylaminease, which was designed by Steve Mayo, which directed evolution took it to make it much faster. And this is really work by Rene and Ricardo, 2 very talented postdocs in my lab. As I told you, in 2013, Don Hilbert and Steve Mayo published a very profound paper where they showed that the original designer enzyme, which is, is called HD3, remember that, which is very, very slow in its catalysis. And I show you here the chemical reaction, this campylaminease, it's an artificial reaction, which there's no natural occurrent enzyme, is catalyzing. So what you need is to break this bond over here, right? To open the ring is a negative base to abstract this proton and a protonated base to stabilize this transition state over here. Okay, and what Steve Mayo and Don Hilbert actually made was a very good transition state analog, which will be very important for the story. Basically mimicking as a stable molecule this transition state. So Designer enzyme speed per second, simple directed evolution, put 17 mutations, these yellow mutations in to make the enzyme to 700 per second. So evolution works. Now comes the dilemma. In the crystal structures, what they saw, here was the designer enzyme. This is a fully evolved enzyme. They're looking identical. That's a transition state analog, except one single residues. So this lysine was designed to be actually the stabilizer of this positive charge, but it was hanging out in nature. And what the directed evolution did, it put this glutamine in, perfectly talking to the atoms over here. Of course, the obvious thing to do, but they didn't do in the paper, is like, oh, the only different one, you don't need the 17, you need this one glutamine. So we made that one mutation, we put in the glutamine, we solved the crystal structure. Perfect. It just looks like the evolved enzyme 700 per second, except when you measure the activity, it's as lousy as the designer enzyme. And that's kind of the story you see for most of these direct evolution papers. The crystal structure looks basically identical to the designer enzyme and is very misleading. So what's the missing link? Haha, we come in with our NMR. The answer must be in the dynamics, right? So let's turn to NMR spectroscopy. So we wanted to see what is the missing link of understanding this 500-fold rate acceleration we get by directed evolution. The first surprise we saw when we took a spectrum of this evolved enzyme, that we give peak duplication. You see here 2 peaks, and the peak duplication got stronger with higher temperature and higher pH, to the point that at high pH and high temperature, we mainly populate the second species. NMR is very powerful because we can assign this peak duplication shown here on red dots. It was really a global process. Second, from the chemical shift, we know it's a second folded species, it's not unfolded. And third, by doing jump experiments, we could show that this interconversion is fully reversible. So we can, shift these equilibrium back and forth. So it's not an unfolding, irreversible aggregation. Now, the other piece of evidence we had was that, in fact, if you look at the activity temperature profile, at higher temperature, it doesn't get more active, it actually gets less active, even so it doesn't unfold, this designer enzyme. So we speculated that second peak is actually an inactive folded species. Now, it turns out this interconversion between this active and inactive folded species is very slow on the NMR timescale. So we could do actually NMR PA jump experiments in real time, where you see that slowly over time, this active species starts to regrow in. Doing the kinetics more quantitatively, we found it takes actually a few hours. So there is an interconversion between two processes. What does it have to do with evolution? It turns out that the original designer enzyme has a much higher population of this inactive species. So the first take-home message, how directed evolution makes enzymes more faster, is actually it selects for an already existing active conformation and increases that population. Okay, you have to have it there. And the designed this active conformation, it was just not there to a high population. Now, in the quest to in the structure of this inactive conformation, we turn to crystallography because it's much faster. And what we actually saw in real time was both conformation, the active and inactive conformation, in our crystal for the APO enzyme. So the reason the other labs had missed it because they only solved the structures in the presence of the transition state analog. And of course, what we found is the inactive conformation clashes with the transition state analog. So it cannot bind. So what the transition state analog binding does, of course, shifts the equilibrium again to the binding competent state. But in the APO form, for both the original designer enzyme and an intermediate, where only 7 mutations were in there and then in direct evolution, do we see both conformation. Now HD3, of course, it's cryo temperatures, we only see the active conformation. One more important information, it turns out, where is this inactive conformation coming from? It was actually in the original scaffold the Mayo lab did for the design, which is xylanase. So it was 100% in the inactive. So in other words, the design could not fully remove this conformation because that was the original very stable fold. But here comes NMR again in why it is so important. these data were actually puzzling because the conformational difference between the inactive and active state was very localized to just 2 strands in the active site. But from NMR, we knew that it's propagating much further out. So my hypothesis was we cannot sample that in the crystal, right? Because the full extent, because it would crack. So we set up crystals at high temperatures and high pH because that's where we should dominate the inactive conformation. And we found something funny, that all the crystals which grew could only grow in the presence of calcium. So now that structure, this calcium, has really large scale conformational changes. You see the active form in yellow, the inactive form in orange. Now we have this full propagation. So to test whether our calcium bound form is now the active conformation, we did NMR on this in the presence of calcium and showed directly that calcium shifts the equilibrium into the inactive conformation. So now we have both conformations and it's really propagated far away, also explaining why the interconversion rate is 2 hours. But then I want to do one more cool experiment, and that's really from the crystallography point of view novel. My postdoc was to build a device that we can do crystallography at 70 degrees. So we are shooting our crystals at 70 degrees, and there it enables the protein directly to interconvert between the both vastly different conformations. So at 70 degrees, we can directly view the active and inactive conformation. Now the last piece of what I have to solve, there's a dilemma. This pre-existing equilibrium gives us only a 3-fold increase in overall catalysis for the evolved enzyme. Where's the other 200-fold coming from? So in other words, this between the designer enzyme and the evolved one, which shifts it more to the active conformation, check mark, we have figured that out 3-fold. And so our hypothesis was the other 300-fold really comes from catalyzing physically the chemical step. How can we get a handle structurally on the chemical step and how this enzyme catalyzes that? And here comes our my good education in enzymology, right? If you all think back about enzymology, we know enzymes catalyze the reaction by lowering the transition state. But the second theory, which much smarter scientists have figured out a long time ago, is because the enzyme has a higher affinity to the substrate in the transition state up on the mountain than the ground state. Now I told you we have this very good transition state analog, right? So we could use that as a directly marker of measuring the affinity to the substrate in the transition state. And here's just the normal equations you know about it, where it came from. The rate of the chemical step is directly related to the free energy to the transition state. And the free energy of the transition state is directly related to the equilibrium constant of measuring that binding to the transition state, either by substate or a transition state analog. So what we did was first titrate in the transition state analog, and we actually could simulate the data. You see, we start with the apoprotein, we get 3 peaks, and at the end, we actually have 2 peaks, meaning we have a conformational selection binding to the transition state, and then an induced pit step. We also, this combined with stop flow experiments gave us all the rate constants, but importantly, the real affinity of the transition state for the different enzymes. And now what you see is that the original and designer enzyme is much, much weaker in its affinity to transition state, about 200-fold than the evolved one. So if you now, based on these equations, plot the rate acceleration during evolution to the increase in affinity to this transition state, it's a perfect line. Why do I tell you that? Because now we know that transition state analog is a perfect mimic to test the transition state, the real transition state of this enzyme. And these are the last 2 slides. What was missing in this in the data? We solved the structures of the transition state analog bound to the enzyme, but again, the answer is in the conformational ensemble and not in static structures. So what I show you here is now ensemble refinement of our crystal structures. Here's the transition state analog, and I want to focus you mainly on the one, remember the one which was very little active, and now the really, really active one. What you just see by eye, the ensemble gets much more narrow. So in other words, the reason why the designer enzymes are so lousy is they are floppy. They are sampling many unproductive states. Yeah, the productive one is in there. Remember, we saw that, but it's just a low. But the excursions to many unproductive states lowers the probability of the reactivity. So the answers in an ensemble. Okay. Now the very last cool thing which Ricardo, my crystallographer, did. I mean, he's just very, very brilliant, very, very creative, right? So he just looked at these data and said, you know, there's 17 residues from here to here, but I guess the key residue is actually this methionine, which gets in the way for an interaction of this in the evolved form of this methionine with a tryptophan. So if you just make this methionine to the cysteine, okay, and the second one, right? So that should actually do the job. So just remember, this glutamine was not good enough because everything is still not well defined. So by just doing this extra single mutation, he recovered almost the entire activity. So that is really good news. We can now have predictive power, right? Second, it's not as magic that you need 17 residues. They just kind of came in there. Only 2 residues were actually essential to lower the transition state. And it's guided by our NMR and crystallography data. So to summarize, nature works. We know that evolution is very powerful. And because, of course, the evolution of power is to evolve function. And the function lies in the energy landscape. So if you are starting to understand better the free energy landscape of the nature evolves world proteins, maybe in the future, we actually have a better chance of designing functional enzymes. In the first story, I told you that a rate-limiting step is a conformational change. And what I haven't told you what we're doing now moving forward, it turns out the rate of my little enzyme is actually rate-limiting for the entire organism. It dictates organismal fitness. So we're doing directed evolution as we speak, where we actually knock out the genomic ADK and put libraries in, and we can actually within days evolve this enzyme to be faster by just survival. I've shown you the really big power of NMR and combining it with high pressure to, for the first time, actually look at structural ensembles of transition states where the rate limiting step is a conformational change and not chemistry, because we can't design transition state analogs for that. And in the second part, and I just gave you a glimpse how we can use the knowledge of directed evolution and really a very promising idea of stabilizing productive substates over all unproductive substates, which is not done currently in the design of designer enzymes. So by implementing that new knowledge, this new concept, we feel pretty confident that we can in the future much better design enzymes. And lastly, I think NMR, and hopefully I've shown you how powerful NMR is to ask these functional questions where I could not come up with a better method which allows you to really look at proteins in action. And so here's just who will be held responsible. It was last year, like actually kind of sort of luckily still for the pandemic a year and a half ago, where we actually had a wedding. That's my biology question. We're kind of productive because grad students in my lab, you know, date each other and get married. And I thank you for your attention.
Is it possible for the inactive species to increase the second peak, that is, even if the protein is kept at a certain temperature over a long period of time?
Good question. It is not. So it's actually, both states are very stable. So it's not that the inactive conformation is kind of aggregation prone or whatever. So the pure equilibrium depends on temperature. That's why we can do the temperature jump, right? You go from low to high and then you're increasing that. But once you have equilibrated, it stays, and pH. So it's not that the inactive conformation is coupled to kind of an irreversible reaction.
Okay, the second question is, so in terms of structural conformation, do you see any remarkable characteristic between the thermal stability and any enzymatic feature?
Yeah, so great question. So of course, I left that out, that first story, right, about the evolution. Our data are very powerful of showing that there is not this trade-off between stability and activity, which is it has been a paradigm which has been floated around for a long time, right? That in order, exactly as the questionnaire is asking, increased stability decreases activity, and that's actually not the case. One of our ancestral proteins, I call it the super enzyme. It is super active and it's super thermostable. And the reason why you don't need this trade-off is exactly the reason why we have big folded proteins. So you need flexibility, which would say less stability in certain regions for this protein hinges, but you can pay for extra stabilities in other parts of the protein. And that's what RNA kinase does. You get increased stability by the core, allowing for flexibility for function. So that's, again, very good news for enzyme design or for biotechnology. Yes, we can make very stable enzymes which are very, very active. Great question.
Okay, next question is, what is the water model used for the solvation? It's not easy to simulate hydrophobic solvation, and this should be necessary for simulating transition states. Do you need to take water polarizability into consideration?
Yeah, I mean, of course, this is a big dream for all of us that we can do using polarized force field for any of those calculations, right? Of course, we have not. I mean, right now the polarized force field are there, but it cannot be done for systems like that. So we use TIP3, and that's why I think it is so important that you combine that with experiments. I think doing it very carefully, and the other thing, the advantage in this system is that we did a comparison. So if it's two homologous proteins, right? So measuring deltas, even by computation, is much more accurate than absolute values, right? But I really, it's not so much the hydrophobic water interactions. The hardest thing where you really need polarizable force fields is actually charged residues interacting with water, as we know, right? And so where we do not use any normal force fields is for chemical steps, meaning breaking bonds and making bonds. But I think for this system, we only believed it really because we had the experimental data I thought we needed to design experiments to test our hypotheses. But it's an excellent question. But in the future, I think we're not far away of computer power to be able to calculate all these reactions and these relations polarizable force fields.
Okay, there are a few more questions. Perhaps you could type answers in because we should move on to Ad's talk at this point. Thank you.
Or we can do that afterwards, right?
Yeah.
Because I want to listen to Ed. I don't want to type.
Of course.
So Rob, it's your floor.
I'm very pleased to introduce what's now the second speaker of this session, Ad Bax from the NIH in Bethesda. Of course, Ad is very well known in this community, but let me try to tell you a few things about his background and career that you may not be familiar with yet. Ad was only 16 years old when he started studying physics at Delft University in the Netherlands. This was in 1972, and he got his master's degree in 78. Not bad for a physicist to become the world's most highly cited chemist, which he was for quite a while. So his PhD research was on 2D, two-dimensional NMR, and presumably inspired by the famous paper by Richard Ernst, which was about 2 years earlier. He got his PhD in 81, again at Delft University, although most of the work had been done in Ray Freeman's lab in Oxford. Both Ray and I were actually in Delft on the jury for his thesis defense. Actually, the thesis was also printed or published rather as a book, probably the first book on 2D NMR. So after postdoc, with Gary Mayfield, he started working in '83 at the NIH, where he still is now, a staggering 38 years later. NIH must have done something well to keep him there all these years. And I mean, not only by paying him a very handsome salary. Let me make one final remark. As you know, Ed was born in Holland, but a couple of years ago, he changed his nationality and became an American citizen. But now, if you look at the incredible political chaos and insanity that you see in the US now, I can well imagine that he is considering to change back. And Ed, be sure that we had no hard feelings, and you're well welcome to become a Dutchman again. So with this, let us proceed with the lecture. The title of this lecture is Transient Structure in IDPs from 3D NUS NMR of Chemical Denaturation. Now, if this sounds a bit mysterious, I can tell you that IDP stands for intrinsically dissolved protein, and NUS for non-uniform sampling. Ed, the floor is yours.
Thanks, Rob. Thanks very much for this very kind introduction. Let me see if I can get the share screen here to work. It's a great pleasure. I also want to thank, of course, the organizers of this tremendous series of lectures for keeping us as a protein NMR community alive and coherent, so to speak. It's a tremendous pleasure to be a part of this series and to have the opportunity to tell you some of the work that's been going on in the group here. And as Rob just explained, the title of my talk says something about transient structure in IDPs. And that's a little bit of a holy grail for some in the NMR community that have been studying IDPs for decades now, particularly the work by Jane Dyson, Peter Wright, and many others in characterizing what is going on with intrinsically disordered proteins that are very difficult to study by other structural biology techniques. So NMR here has a unique opportunity to contribute new knowledge. It's proven to be very difficult, though, to do this. And we've been engaged, basically people in my group have pursued this for a while, looking at synuclein, A-beta, and several other proteins. And one of the ideas is that, and that came out of Peter Wright's work and other groups, there should be transient ordered elements in those structures. But when you look at what's come out of the NMR community, mostly what you see are basically uncooked or cooked spaghetti kind of models for proteins that, from my perspective, are not particularly informative. So I was interested in, is it possible to find potentially well-ordered transient elements in an IDP that would, according to Peter Wright and others, be important for function, and the importance, of course, being highlighted by the fact that those conserved regions, that those regions that are disordered are highly conserved in nature. So this happened all just before the pandemic, that I got 3 very young, bright guys that shared the same office, and this new guy, Tayyip, came and asked for a project, and I said, well, you can look at IDPs because they give great NMR spectra, and at least you get your hands wet. He'd never done a protein NMR, recorded the protein NMR spectrum before, so I said, at least you'll get great quality spectra. And that's just when the pandemic happened, so I lost sort of sight of those guys, but they must have talked to one another. And I'm also somewhat of a conspiracy theorist, that of course, living in DC, you become that way. And I think those 3 of them got together and they said, let's go and prove that the boss was wrong. And I'll show you in the next slide what I'm going to be talking about. Now, mind you, they did get assistance of 2 very bright staff scientists in the group. Yang Xian here, seen in the glow of my blue laser that he right now plays a key role in, but known to many of you as Mr. Talos or Mr. Sparta, the programs that he developed among many others, and Mr. Super Smile, Jin Fa Ying here, that, of course, you know from his work on non-uniform sampling, and that plays a key role in the project I'll be talking about. Now, when I said the conspiracy theory that they want to prove the boss was wrong, were we wrong? They picked a system, a beta, that about a half dozen years ago, Julien Roche and myself had published a biochemistry paper on that showed that the chemical shifts here, the secondary chemical shifts, are very close to random coil. A beta 42, which is very amyloidogenic, and the A beta 40, which is less likely to form or less prone to form amyloids, have identical chemical shifts in solution, right? Except for half a dozen C-terminal residues. So chemical shifts being random coil, J couplings being close to random coil, chemical shifts being the same between this highly fibril forming form and the much less amyloidogenic form. I mean, what is there to do pursuing NMR? You look at the NMR spectrum, the chemical shifts are right on top of one another, except for here G38, glycine 38, close to the C-terminus. Obviously, this thing is 40 residues long or 42 residues. Differs a little bit. 37, interestingly, differs more. A couple of ppm in nitrogen, but nitrogen is very sensitive, so what the heck? It's a C-terminus, who cares? Now, of course, this system is being studied to death by others, molecular dynamic simulations, NMR spectra. There's one interesting paper that I sort of discarded at the time, and I should have paid more attention to, by Yan and Chunju Wang, that looked at relaxation, they did spectral density mapping, and they said, well, this thing at the very C-terminus, 42 is a little bit more rigid than 40. And at the time I sort of like, well, yes, duh, it's a little bit longer, not that interesting. Now, Julien, when he recorded data, what he saw was actually something interesting that we sort of shoved under the rug, but it's a figure in the paper that he published, is that there is a tremendous change or differentiation of backbone dynamics. If you look at the zero frequency spectral density, and that's reflected here in the N15 trend T2, it differs by about a factor of 3 depending on chain position, and that correlates with the buildup rate of intra-residue amide alpha NOE, all right? So this thing is not just equally flexible, uncooked spaghetti. It looks like there is some parts that are more ordered than other parts. And in my defense to the 3 postdocs, we did point out at the time that the chemical shift data strongly suggest, but do not conclusively prove, that the population of any ordered structural element is very small. Well, and that part turns out to be correct. The population of the ordered element is small, but presumably I'm going to be hopefully going to be able to convince you it may actually be relevant to the biology of this A beta peptide. And the population is going to be about 10%, and it's going to be beta sheet. Now, of course, many of you know about IDPs and IDRs and this spaghetti model. And this is just many of mammalian proteins, about 30% or so, have those big disordered regions that are totally disordered. You know, you got molten globules that supposedly behave like highly flexible. The idea is that there are transiently formed structures, but mostly they're being thought of by people as random coils. But the question then that the guys wanted to address, can these transiently formed structures, if they do form, be important for protein function? And Peter Wright, Jane Dyson, and others have said the answer is yes. Now, does A beta have such a structure? I'll show you that the answer is yes, and that it's a beta sheet, and that it's sensitive to mutation, oxidation, and other factors, all right, that are known to be related to fibril formation propensity. Now, when I say it's a new technique, we call it TSD NMR, as I showed on the title slide. Sometimes we joke there is nothing really new in NMR since Abraham published his textbook in 1960s. And along the same lines, there's nothing new in protein NMR that Fleming Paulson, the late Fleming Paulson, he unfortunately passed away untimely about 8 or 9 years ago, I remember. There's very little new in NMR that Fleming hasn't already touched or worked on. And the same applies to the technology I'll be talking about today. Others have expanded on this, others, in particular Robert Conrad and various others. And this is the use of urea to highlight the presence of transiently ordered elements in a what would be considered a disordered structure. And Fleming here looked at acyl carrier binding protein in the acid denated state, but it wasn't totally denated because when he added urea up to 5 molar, you see the chemical shift changes here in the direction of random coil from the conditions that he had at 2.3 pH. And here you see another element, and he says those are helices that were still transiently present. All right, and the chemical shift changes here is about 1 ppm, so there must have been about 30% transient helix, cooperatively, mind you, present in this acid denated state, a little bit less here. And most of the other examples that you see from Robert Conrad and others focus on helices. We're going to be looking at beta sheet, which turns out to be a little bit harder perhaps, and at populations that are much lower than 30%. All right. So it turns out to be a little bit of a challenge, but it turns out to be doable. And the theory, if you want to call it theory, it's very pedestrian thermodynamics. It's already been discussed at length in Dorothee's talk, so I'm not going to really dwell on it, but we're looking at this IDP as an equilibrium between a folded and an unfolded state. But for normal proteins, of course, this is populated, and the unfolded state may be 10 to the minus 3, 10 to the minus 4, or 10 to the minus 5 populated. Here, for an IDP, is the unfolded state that is highly populated. So if we look at the unfolding diagram as a function of the urea concentration, the red protein would be a normal protein that's highly stable, that unfolds with a cooperativity that's typically expressed as an M factor. And here you can see the ratio between folded and unfolded population. We write it as an equilibrium constant times exponential minus M times the urea concentration divided by RT. Okay, so we've got this equilibrium. We can shift the equilibrium by adding urea and get the unfolded state to be even more populated. Now for an IDP, we're somewhere here, all right, that we're shifting from a protein that is slightly folded, transiently folded, to when you add more urea, becoming more and more unfolded. Okay, you can write that again in very simple equations. And importantly, if the exchange rate between the folded and unfolded state is fast, any chemical shift we measure is going to be the average chemical shift between the transiently folded state and the unfolded state. Okay, so fast exchange is a precondition, and we get a weighted chemical shift difference between the folded and the unfolded state. Now, the buried surface area in the folded state is related to this cooperativity of unfolding. This is an empirical relation that's been developed 20 years ago by Schultz and others. And of course, if you're going to have a small nucleus that is folded, this cooperativity is going to be small. Okay, so the equilibrium constant, this would be at 0 mole urea, the ratio between folded and unfolded. We call that A. It's a little bit different from what people normally do, but it's adapted here for the IDP story. M depends on the protein and the denaturant. Okay, so we could use any denaturant, but we're going to use urea because it's very NMR friendly. Now, one more thing is that since we're looking at a protein that is highly unfolded, only a very small fraction is transiently folded. If you look at this unfolding curve, all we've got here is this tail of what normally would be the normal unfolding curve, all right? So we use a very small M value for this simulated graph here, this calculated graph, M of 0.2. It turns out for A beta, we're going to have an M of 0.06, corresponding to about 400 square angstrom of buried solvent accessible surface area in the folded state. And the A, here it's shown for 0.3, the A is going to be about 0.1 for A beta. Nevertheless, I'll show you that this may actually be quite well relevant. Now, the tricky bit is going to be that we only got this little curve here, and this is already exaggerated because the population that we're going to be looking for is lower. And you're looking at this tail here, you can express this chemical shift then as the unfolded chemical shift, the fraction of folded chemical shift at 0 mole urea, and then the difference between folded and unfolded with an exponential minus m times the concentration of your urea. Okay, so very trivial. Now, you look at this equation, obviously what you got here is this m is the curvature of this blue line. It's very little, so it's not that easy to get that out precisely and accurately. The other thing that you can see from this equation is that the only thing you're ever going to get is the product of A, that's the population, and the chemical shift difference of the folded and the unfolded state. All right, so you cannot uniquely determine the population of your folded state. But we can use a little bit of common sense, because we know that folded states have secondary chemical shifts. For example, Wishart's RCI order parameter, the random coil index, is based on the fact that all folded proteins have roughly comparable secondary chemical shifts. So we can assume that whatever folded state must have on average roughly secondary chemical shift. There are a couple of ppm for C alpha, C prime, maybe 4 or 5 ppm for nitrogen, roughly, right? And it turns out that the spread in folded proteins on average is very small. So we can use that as an additional restraint when fitting our data, that we don't want to get crazy secondary chemical shifts. So we constrain the secondary chemical shifts to be roughly reasonable. And we don't do that for just C alpha, we take the whole lot of them together. So we can make small C alpha chemical shifts as long as the nitrogen secondary chemical shift is large, or vice versa. So we're comparing all the secondary chemical shifts, using it as a single restraint term to prevent the problem from diverging when you're searching for solutions. All right. Now, this is a slide looking at an HSQC titration of this A beta 42 and A beta 40 peptides, where we're just zooming in on glycine 37 and glycine 38 regions of the HSQC spectrum. And lo and behold, what you see here is that for A beta 42, I showed you already earlier, the chemical shifts are rather different from A beta 37. That's the green one here, or let's focus the purple one. Green is the oxidized version of it, methionine 35 oxidized version. You see that for glycine 38 and A beta 40 goes upfield when you add urea. For A beta 1 to 42, we move downfield when we add 8 molar urea. And if you go to infinite amounts of urea, of course, practically not feasible, you see that those guys are converging somewhere around here. Now, we have to worry about a solvent effect as well, because we're changing the solvent conditions when we add 8 mol urea. And there's going to be urea that aids hydrogen bonds to the peptide group that may have some impact on the chemical shift. So we can also ask the question, what does it do if we make a short synthetic peptide of no structure, but the same amino acid composition for the 3 residues? So glycine 38, its preceding residue, its following residue, and I'll come back to that in a second. So we look for a tripeptide and see what's happening. And for the tripeptide, very little happens. So we're looking here at the difference in chemical shift behavior that is related to the unfolding of transient structure, as I'll show you in a second. Here you see the same thing for glycine 37. You get large changes in chemical shift. For A beta 42, smaller changes in chemical shift for the blue guy, the oxidized form that is less amyloidogenic than A beta 1 to 40 wild type. Okay, this is the methionine 35 oxidized. And the same is true for A beta 40 and A beta 40 oxidized. So those guys converge when we add enough urea. And we can do the same thing. Of course, just protein and nitrogen are not structurally very informative, but we can get the CO, the carbonyl, and the C alpha chemical shifts by recording triple resonance spectra, because those small proteins or small disordered peptides give great NMR spectra. And in a matter as little as an hour, we can get an HNCO and an HNCO-CA spectrum by using non-uniform sampling. It needs to be done fast, because if you go to high urea concentration, you get problems like carbamylation, a chemical reaction occurring. And A beta peptides, they can fibrilize within hours in the NMR tube. And of course, we're going to be relying on non-uniform sampling to get the most accurate possible peak position of those resonances, because the chemical shifts are going to be exquisitely important for the analysis that we're going to be trying to do. The peptide, of course, we're using those reference peptides, synthetic peptides. We can't do 3D HNCO or HNCO-CA on it because they're natural abundance. So those are just recorded 1D spectra, trivial reference data. Now, I mentioned the solvent effect, and that is a bit of a bottleneck in this work. It's a really nasty property that urea has different effects on the chemical shift of lysine versus isoleucine versus alanine versus, say, glycine or valine, even when it's embedded in an environment of glutamines. We pick glutamine here following Fleming-Paulsen, who says this is the most average residue of any residue that you can pick in terms of being innocuous. It's not particularly hydrophobic, not hydrophilic. It's basically an average residue, he said. And we like that idea, and it functions very well for this purpose. But what you can see is that here, focusing on the N15 shift, but all the other chemical shifts behave the same way. The different residues change their chemical shift to different extents, even while they're totally disordered because of the effect of urea. So you got to factor that out if you want to interpret chemical shift changes when changing the urea concentration. Now, what Tayyip and Venkat did was they said, well, we're looking here at the C-terminal sequence of A-beta 42. They made pentapeptides, and say for isoleucine 32 here, they say we make a peptide, glutamine, isoleucine 31, isoleucine 32, glycine 33, and another glutamine, to see how this residue behaves if we just change the solvent by adding urea. We're going to do the same thing, make a new pentapeptide. Sorry, I left one out here. Make a new pentapeptide here. There's a glutamine here at the end as well, and see how this leucine behaves when we add, and then compare that to it behaves in the native A-beta peptide. All right, so it's a fair bit of work in terms of peptide synthesis and recording those trivial 1D spectra as well, and doing the urea titrations. Oh, that keeps the guys busy in the lab. That's good, because I didn't have time to look after them. This is what they came back with, and showing how the chemical shifts change as a function of urea concentration here, molar concentration of urea. And what you can see, they all converge to the same values. These are now corrected for the effect of urea on the pentapeptide, disordered pentapeptides, and they nicely converge. So they can fit this to this equation using the chi-square function that I've shown you earlier, saying that the chemical shift is an average between the folded and the unfolded states. And from that, importantly, they can get the chemical shifts of the transiently folded state. And they get the population in unitless units here. This is the fraction of folded versus unfolded conformer. And as you can see here, for the A-beta 1 to 42, it's 0.11. That corresponds to 10% folded structure. It's less folded, transiently folded, for the less amyloid forming A-beta 142 oxidized. It is identical if we change another residue, lysine 28, that's involved in putative salt bridge formation, to 22, aspartate 22 or glutamate 23. Now, the cooperativity that they fit with this equation comes in at roughly 0.06 kcals per mole, or the 400 square angstrom buried surface area that I mentioned to you earlier. Now, interestingly, those folded chemical shifts have unique signatures. We see some large secondary chemical shifts, in particular when looking at the nitrogen chemical shifts of A-beta 42, of those glycine 37 and glycine 38. Remember, those were the ones that were changing a lot. The same for the carbonyls. We see large chemical shift differences for 33 and 34, 38 and 39, and the same here for the C-alpha chemical shifts. There are rather substantial chemical shifts. And then the trivial thing to do, well, trivial, if you have Yang Shen in your lab, the guy that developed Sparta and Talos, he just searched the database and says, give me the chemical shifts and I'll see, is there any decapeptide in the database of crystal structures that have NMR chemical shifts that matches the chemical shifts you gave me. And lo and behold, when he searched the database, 8 out of the top 10 hits are type 1' beta turn structures, and they're superimposed here. One of them turned out to be an extended structure. I bet you that this one actually was transiently folded in the NMR tube, but just in the X-ray structure, it did not. It was a C-terminal sequence. It didn't adopt the beta turn. It wasn't reported in the X-ray as being a beta turn, but it may well actually, considering the chemical shifts being so homologous to all the other ones, have formed this type 1 beta turn. Now, I was a little bit skeptical because I said, chemical shift modeling, you know, it's modeling is modeling. Do we have any more evidence? And I wasn't particularly involved with this project, but Jinfa Ying said, hey, remember, we published this paper a couple of years ago showing that NUS, you're better off using time-ordered sampling when doing NUS experiments, when your sample isn't very stable. And what those guys did to highlight that method was they looked at 1.2 millimole of A beta 1 to 42 that would normally fibrilize in a fraction of a second, but they did it at 3 kilobar and at high pH. And at 3 kilobar, they could keep the sample sort of alive for about 24 hours. So while it was decaying, it decayed about, there was 25% left at the end of the experiment, but they got incredible signal to noise NOESY spectra. So they would be able to see a small fraction of folded state in the NOESY spectrum. And indeed, he was able to see a long-range NOE, or officially a medium-range NOE, between valine 39 and valine 36 amide protons. And equally, and perhaps even more surprising, and I was rather suspicious at the time, a long-range NOE between the amide of isoleucine 41 and methionine 35 H-alpha. All right, so those are 6 residues apart. I gave him a hard time over it. I said, well, how do we know it's not an NUS artifact? Use different sampling protocols, reran it. So this experiment got done 3 times, and every time this little peak showed up. So I couldn't deny it was real. Those were the only 2 NOEs that were unambiguous, not expected in sort of a transient, very small turn or whatever. So those are the only ones that we actually included in the paper at the time. And these are actually in total agreement with the structure that was modeled by Yang Xian. Another way of confirming it is by doing PRE experiments, because of course you all know that PREs, this is Marcus Weckstetter's experiment with Bertoncini, showed for synuclein, for example, that tagging an IDP with an MTSL is fantastic for seeing transient structures. That is not just a random coil, you see transient interactions occurring. And what we're seeing here is when we put an MTSL tag at the C-terminus of this peptide, that we see interactions. We see that the paramagnetic species, the black spectrum, we see that 38, very close to the C-terminus, very close to the tag, stays pretty good in intensity. Glycine 9, very far away, stays good in intensity, but 29 gets weak, 37 gets weaker, and 36 and 35, 34, and 33 totally disappear. All right, so we do have long-range interactions, or at least not just immediate proximate interactions. And what you see here is the ratio of intensities in the HSQC spectrum when the tag is here at the C-terminus. So the immediate C-terminal residues, of course, are attenuated, but then here from 39 to 36, the intensities are preserved, and then 35, 34, 33, and 32 are attenuated below the signal-to-noise threshold. And we see attenuation elsewhere in the peptide as well. We add, importantly, we add urea to remove this transient structure, and the intensity here comes back up. And here you see the agreement with the 1 over R to the 6 distances that are modeled in this structure, and the NOEs are consistent with this as well. So I hope that you believe that this is indeed some independent validation. The structure, the buried surface area in those peptides that came out of Yang's database search was 370 square angstrom, very compatible with the M value that Venkat and Tayeb determined from their chemical shift fitting. Now, I want to end my talk with a non-NMR slide. I consider this a public service message of some of the stuff that you may have seen various times. during the pandemic that got highlighted in some news stories. It's a public service message, sort of jokingly say, you got to realize that everybody, when we speak, we spit out saliva, and we know that saliva contains respiratory virus, such as COVID-19 or influenza. And here you can see how much is actually spraying out of my colleague's mouth when he speaks in front of a very bright light sheet of laser light.
Spit happens.
See, he says spit happens.
Spit happens.
Everybody,
Spit happens.
even I spit.
Spit happens.
And
spit happens.
This was the youngest,
happens.
most enthusiastic member of our team, spitting droplets through the sheet. And it's important to remember, we all do this when we speak. Those droplets keep hanging in the air, not for seconds, but for minutes or a good fraction of an hour, not for hours, but for a fraction of an hour. So there is no shield, there's no plexiglass that's going to help you. The only thing that's going to help you from getting those things in the air is to wear a mask. It can be any mask. You see, even a paper towel is a very effective way of stopping the virus from getting out into the atmosphere. It may prevent you as well from inhaling it. That's much harder because then the droplets are much smaller after they evaporate the water fraction of it. So those things rapidly shrink, but the virus stays alive and is actually remains viable when it's fully dehydrated. So when you lyophilize those droplets, or you put them in a 30% relative humidity atmosphere, they live essentially for almost forever. So they drop to the ground before they're dead. So there's a lot more that you can learn on this. I gave an APS webinar with some of this, the background on it. It actually has an NMR slide in there, but it's an hour and a half of tedious discussions. But in case you wanted to know more about that, you can look it up there. At this point, thanks for your interest. And if there is any time left, I'd be happy to answer any questions.
Thank you, Ed, for a beautiful talk. I have a few questions from the audience. Let's see. Dubini, he asks, how can one be sure to differentiate between the structuring phenomenon occurring in the monomeric level versus an artifact due to the oligomeric photo-aggregated but still detectable species for beta samples?
Yeah, no, it's actually a very good point, because there's been many structural results published, and we know that we could have, especially at high concentrations, everybody likes to work for NMR. You don't like to do this at nanomolar, you like to do this at micromolar, let's say 100 micromolar or whatever. So you got to be 100% sure that you're not looking at a transferred NOE effect, for example, right? So we did try to make that 100% sure by looking at concentration dependence. So there's zero concentration dependence of the chemical shifts. So the chemical shifts that we have are really the most sensitive indicator of transient structure. The other thing, and that's been published not by us, but by Nick Fawzi and Marius Clore, when Nick was still in Marius's group, they paramagnetically tagged A beta in a mixed form. So they used unlabeled, tagged, mix it with labeled, N15 labeled, untagged, and see is there any kind of paramagnetic crosstalk, and there was zero. All right, so we've looked for that pretty hard, and there is no evidence for transferred NOE effects or for transient oligomerization under those conditions. But it's a very good question, and I think under different conditions you can get those transient aggregation effects to occur, and those may have been well, I wouldn't say to blame, but responsible for some of the findings, experimental findings by others.
There's another question by Frans Mulder. Can we compute M from sequence?
Can we compute M from sequence? No, well, you can't compute it from a random coil sequence, but you can compute it, of course, from structure. It's very hard to predict what kind of M value to expect. You need a structural model. So if you have a structural model, you can sort of calculate standard methods. We used a very primitive program, but they all gave the same answer. What is the buried solvent accessible surface area by comparing it to a fully extended structure? And that was a sanity check to see whether this element that we came up with had actually a reasonable answer. And perhaps it's almost too good to be true. It was smack on the money of what the empirical equations predict.
Let's see, a question by Supriya. How would the CS exchange model or equation look if there is a third state present?
Yeah, again, a very good question. We cannot exclude the presence of a third state. Of course, it gets more complex. It's really Occam's razor that we can fit the data with the two-state model. Of course, nothing is totally two-state because there has to be a path to go from one state to the other, as Dorothee so nicely explained. You got a transition state, you got a path to the transition state, you got something on the other side. So it's Occam's razor. It's the simplest model to describe a state that is disordered and transiently ordered. There are other transiently ordered states in this peptide, but they seem to be not correlated to the C-terminal beta hairpin. We can tell from the paramagnetic states that there are transient interactions to the N-terminal region, for example, but those are not cooperative with the beta sheet formation. And for that, we did this K28E mutation, and we made some other mutations. So that's part that I didn't discuss in my talk. But yes, real life is more complex than the simple thing that I just gave you. All I wanted to do was illustrate that this technology of urea denaturation and very careful chemical shift analysis allows you to identify unambiguously, uniquely structural elements in IDP that presumably are biologically relevant.
Okay, there's a question by Guy Monteleone. Would you expect the increased pressure to unfold the transient structure? Are the NOE's effects you see perhaps pressure dependent?
It's a very interesting question, Guy. I can't really give you an answer for this A beta. The pressure dependence of such very small structured, buried structure, small elements is way too small. We would have to go to 50 kilobar instead of to 3 kilobar to be able to see a reasonable pressure dependence for those very small buried surface areas. So my answer is we can calculate it probably, but we can't really easily observe it. We see that some transient helices are pressure dependent, other ones are not. Helix packing is pressure dependent, individual helices to a much lesser extent. So yes, there's a lot more that NMR can tell us with more careful experimenting. There's many, many years for many, many groups to be done here. So that's my preliminary answer. There's really a wealth of information to be uncovered.
Okay, well, I can go on until I see a sign that I have to stop. Huh? So, okay, by Fleming Hansen. And do you know the rate constant between the random coil state and the beta hairpin structure?
Yeah, great question, Fleming. I love that question, and I've been pestering the guys, trying to keep them busy while I do other stuff and giving them a hard time. Can we see what's the rate? What's the lifetime of my transiently folded state, right? Is it milliseconds? No, because we would see exchange broadening. Is it microseconds? Well, it has to be longer than about 10 it has to be shorter than 10 microseconds, because otherwise we would have seen an effect on the Trosi R2 of the N15 line width. So we looked at it pretty hard at 900 megahertz, and we just barely start seeing perhaps an inkling of a little bit of broadening of those two glycines that have the largest chemical shift change in the N15 dimension between folded and unfolded states. So it looks like it's low microseconds or high nanoseconds, but I don't, the data are so like just so borderline. We need the 1.2 gigahertz to really get an unambiguous answer for this question, but great question.
Okay, one by Remco Sprangers. Thanks a lot for a great talk. Why are the PRE effects around residue 36, 37 not fully gone in 8 molar urea?
Yeah, Remco, again, a great question. But remember those chemical shift titration curves that we were looking at, 8 molar wasn't enough to fully denature the peptide, that transiently folded structure. 8 molar times 0.06 is 0.5, right? So the exponent in my exponential is exponential minus 0.5. So we're only half denaturing, a little bit more than half denaturing our transiently folded state. What we need is guanidine that should be able to do a better job, but doing NMR in 8 molar guanidine is not so easy. But then we should be able to get another factor 2 denaturation.
I would like to ask one myself. About NUS, I always understood that the main advantage is to have more sensitivity because you sample the strong part of the FID more than the weak. You have used other advantages, or you find other advantages of this method?
Yeah, it's an important question, Rob, and thanks for being able to sort of highlight that this really is a misconception. You can use NUS perhaps for a marginal increase in signal to noise. And if you look at Jinfa Ying's SMILE paper, we sort of make a big point of this. Yes, by sampling at the beginning, you can increase signal to noise somewhat, but it really would go at the expense of intrinsic precision of your peak position. So here, what we really need is we need the most accurate possible peak position we can ever get in the minimum amount of time. We're not using it for signal to noise, getting a little bit signal to noise enhancement. What we need it for is we need it getting that 3D spectrum in less than an hour before carbamylation and precipitation of our peptide kicks in. All right, so we'd like to use the same sample and just add urea to it so we don't change anything. And so it needs to be done fast. So it's done here for speed. So we can record 8 spectra in 24 hours or less, 16 spectra, 3D spectra, 8-NCO and 8-NCO-CA. So those spectra, each of them was basically an hour. And it's important, those IDPs, even at 100 micromole, you can easily do that.
Okay, I think I take one more. Can we use PFG NMR to estimate the transient structure of IDPs by estimating their effective dimension during urea titration by Salikov?
By using, what was the last thing? By
no, that was the name of the guy who, Vladislav.
Okay.
Sorry.
I presume that the idea is to look at diffusion rates, perhaps. It'd be very difficult to get the kind of precision accuracy that you would need to see a 10% population of something that is compact, that is averaging with the diffusion of something that is much larger, that diffuses like a cloud, that is behaving like it's 5 times the size, right? So I'm not optimistic. It's not fundamentally impossible, but in practice, that's tough going for those very small populations of transiently structured elements.
Okay, I think we should stop here because there are some questions left, but we're losing the audience at a scary rate at the moment. So thanks a lot, Ed, for a really beautiful talk. And